Hello everyone and welcome to your first virtual lecture for proteomics. So what we're going to try to cover in this set of videos, with each one of these topics in this checklist being its own separate video so that you can kind of take this at your own steady pace. What we are going to try to cover first in this video is the basic makeup of proteins, especially since proteomics is essentially the science of studying proteins, so it's not really going to be a surprise that a lot of our focus this semester is going to be on proteins. So we are going to learn about amino acids, the constituents that make up proteins. We'll learn about how amino acids are joined together to make peptides of increasing length until you finally get a protein, and we'll learn about the peptide bonds that actually make up the cohesive bonds that join amino acids together. And then later videos will focus on things like reviewing how proteins are made in the cell, so you likely learn this not only in your introductory biology class, but also in Intro to Biotechnology. We'll talk about how proteins are folded. That'll be a little bit of review as well. We'll talk about the evolutionary relationships between proteins. That should be something kind of new and exciting for us to talk about. And then lastly, we'll have a brief introduction to different classes of proteins and what we can kind of expect their functions to be. Something that we'll talk about more and more as we make our way through the semester. So for now, let's go ahead and tackle the first topic on this checklist and start talking about how proteins are actually made up of amino acids. So the fact that a protein is a polymer of individual units, in this case amino acids, that is not something that is unique to proteins. So all biomolecules like proteins or like nucleic acids or like polysaccharides, they're all polymers that are constructed in basically the same way. So the cell is going to build proteins or build nucleic acids by using enzymes to catalyze dehydration reactions to join monomer units together. So this is just kind of the basic representation of what a polymerization reaction looks like. So you have monomer number 1 here, monomer number 2 here, and the reason why we call this a dehydration reaction is because in the process of forging a covalent bond between the two monomers so that they're no longer separate, we lose an element of water. You'll notice that if you look at the two hydroxyl functional groups here on each of the monomers, you'll see outlined in blue here OHH. Well, that's H2O, so when we form that covalent bond, you'll notice that the OHH is no longer present. So that is why we call it a dehydration reaction. We lose the element of water in the process. So this is just kind of a generic representation of what a polymerization reaction looks like. So what's going to separate one kind of polymer from another, so say a protein from a nucleic acid, is going to be the identity of the monomer that is used in the actual reaction. So what is the monomer that actually makes up the links in the chain? So in the case of a protein, we are going to learn about this a lot in this chapter. Proteins are made up of individual amino acids that are joined together by dehydration synthesis. Nucleic acids like DNA and RNA do not use amino acids. They use something else called nucleotides. Carbohydrates use monosaccharides, and then lipids, the final type of biomolecule, doesn't really fit into this polymerization concept. It's going to kind of vary from lipid to lipid, so you don't really have to worry too much about lipids in that regard. But our major takeaway here, if we're going to focus on proteins for this proteomics class, your major takeaway should be a protein is a polymer that is made up of individual amino acids. And the name of the covalent bond that is formed for each dehydration synthesis reaction is called a peptide bond. So let's look at what your standard generic amino acid actually looks like. So we already know proteins are polymers of amino acids. Okay, so here is what makes proteins so complex and so varied. There are 20 different kinds of amino acids that we can use to make proteins. So if you think about, say, the alphabet, there are 26 letters in the alphabet, and we can build words by sticking together letters in a number of different combinations. So the word proteomics, Proteomics, contains a few of the 26 letters that are put together in a certain way so that it comes across as a understandable, legible word in the English language. Proteins are the same way. We can make a big, long polymer of amino acids when we're making a protein by sticking any of these 20 amino acids in any order that we desire. So there are 20 different types of amino acids that we can use to make proteins. So something that all 20 of those amino acids have in common, if you look at this amino acid here in terms of its chemical structure, all amino acids have what's called a central alpha carbon. That's this carbon right here. And then attached to that central alpha carbon, we have a basic amine functional group. It's colored here in blue. We have an acidic carboxyl functional group here colored in red. And then we have a variable R group, or what is more appropriately called a side chain, which is colored here in orange. So all 20 of the amino acids are going to look pretty much identical to what you see here, but what makes one amino acid different from another amino acid is the identity of that side chain. The letter R here does not mean some element on the periodic table that has the atomic symbol R, because no such element exists. The R basically means remainder. It's the remainder of the molecule, which is kind of shown as R as kind of a means of shorthand. Some of the side chains can be rather large and rather bulky and contain a lot of atoms, so sometimes it's just easier to just say, okay, I'm going to skip all that and I'm just going to write the letter R. But we are going to want to pay attention to what makes the different side chains unique and different from one another, because that is what makes an amino acid unique here. So as we said, when you are joining amino acids together, you are utilizing the process of dehydration synthesis, which is going to be catalyzed by enzymes. So the peptide bond is the name of the covalent bond that is forged between the two amino acids. And this happens between the amine group of one amino acid and the carboxyl group of another amino acid. So if you look at this diagram here, you'll notice that we have two different amino acids here. So this is your first amino acid here on the left. This is your second amino acid here on the right. So the way we have these two molecules juxtaposed with each other is we have the carboxyl group of this amino acid right next to the amine group of this amino acid. So if you look here in red, you'll notice again the element of water that is being highlighted here, HHO, H2O. So that once we actually form the chemical bond here, once we actually form the peptide bond, the element of water is lost, and now you actually have a chemical bond being formed between the carbon of this carboxyl group here and the nitrogen of this amine group here. So that's your peptide bond right there. So basically you get rid of those atoms that are colored in red, and you attach the C to the N right there. Attach the carbon to the nitrogen. That is the peptide bond. And like I said, the net loss of a water molecule here is why we call this a dehydration or condensation reaction. So once you join two amino acids together like this, we call the resulting molecule a dipeptide. Di being the prefix meaning two, and peptide, of course, referring to the peptide itself. Dipeptide. You could then have a tripeptide, a quadripeptide, and so on and so forth. So we can continue to add amino acids again and again and again. We can add amino acids to the dipeptide to make a tripeptide. We can add another amino acid again and again and again. We can theoretically make this polypeptide polymer as long as we want it. And that's going to be what we end up doing when we get into the business of making proteins. You'll notice here that if you look at this tripeptide that you see right here, so to help you out, they're labeled 1, 2, and 3. So this is your first amino acid. There's the peptide bond that joins it to the second amino acid. There's the peptide bond that joins it to the third amino acid. So if you look at this tripeptide here, I want you to notice a couple of things. The first is that no matter how many amino acids we add to the chain here, this peptide, this tripeptide or polypeptide or whatever you want to call it, it is always going to have an exposed amine group and an exposed carboxyl group. Now, it is true that we did lose an exposed carboxyl and amine group in the process of forming a peptide bond, but we are always going to have one left over on each end of the polymer. So these exposed ends are what we call the N-terminus if we're talking about the free amino group, and the C-terminus if we're talking about the exposed carboxyl group on the opposite end of the molecule. So no matter whether we're dealing with a dipeptide that contains 2 amino acids or a very long protein that contains hundreds of amino acids, each one of them is going to have an N-terminus on one end and a C-terminus on the other end. The other thing I want you to notice here is look at the side chains, which are being represented as R, R prime, and R double prime. So each amino acid, even as the amino acid is incorporated into a peptide polymer, that side chain is exposed and just kind of hanging out all by itself. And the reason for that is because the side chains do not actually participate in the chemical reaction that results in the formation of a peptide bond. Since they are not participants, they are just going to be kind of hanging out to the side here. So the most important thing to understand about the side chains once their amino acid is a part of a protein or a peptide is that these side chains have some freedom to be able to interact with other molecules in the general vicinity, other molecules that might come into contact with the protein or the peptide, like water, for instance. Okay, so look at this diagram here. So obviously we have a peptide of some undetermined length here. So somewhere over here on the left we have the N-terminus, somewhere over here on the right we have the C-terminus. So we're just kind of focusing on the backbone of this peptide as you go from alpha carbon, carboxyl carbon, peptide bond to amino, nitrogen, alpha carbon, and so on and so forth. So we've got one amino acid here, one amino acid here, and so on and so forth. So what we want to talk about here is the concept of rotational freedom. So atoms that are connected by single covalent bonds, a topic that you probably remember from general chemistry, a single covalent bond referring to the sharing of 2 electrons between 2 atoms. Well, atoms that are connected by a single covalent bond typically have what's called full rotational freedom, meaning that that bond can rotate a full 360 degrees and therefore adjust the position of the atoms that are attached to it. So the reason why in a lot of cases you want bonds to have rotational freedom is it allows those atoms to rotate until they find a state of highest entropy, which is in line with the second law of thermodynamics, which says that all physical processes proceed such that the level of disorder in the system reaches its maximum. So basically what that means is that we try to rotate these bonds so that the atoms attached get as far away from each other as possible. So there are a number of different chemical bonds that actually make up a peptide. So you've got the bond between the alpha carbon and the carboxyl carbon. You've got the bond between the carbon and the nitrogen here, the nitrogen and the alpha carbon. So we've got a whole bunch of bonds here. So not every bond that we see here is actually a single bond. It turns out that the peptide bond that joins the carboxyl carbon to the amine nitrogen is actually not really a single bond. It actually has some double bond characteristics. So a double bond actually shares 4 electrons rather than 2. So because the peptide bond has some double bond characteristics, it's not going to have really much rotational freedom, meaning it's going to be a lot more rigid. So you're not really going to be able to rotate that bond 360 degrees like you would other bonds in the peptide structure. So because of the rigidity here, basically you can kind of think of the carboxyl carbon and the amine nitrogen as being kind of fixed in a plane, which is why in this diagram you kind of see these little rectangles here, which are meant to indicate that the carbon and the nitrogen here are basically stuck in plane with each other, that there's no rotation there, so they're basically stuck in that particular configuration. Now, where you will see some rotational freedom here is between the alpha carbon and the carboxyl carbon, so you can see a little arrow here indicating that we do have rotational freedom there. And then you will see some rotational freedom between the alpha carbon and the previous amino nitrogen. So you do see the arrow there indicating we do have rotational freedom there. So the angle of rotation for each of those is indicated by these Greek letters here, both theta and omega here. Or, not omega, psi. So theta and psi are indicators of the angle of rotation that you get for those particular chemical bonds, but we don't see that for the peptide bond, as we said, because there is no rotational freedom there. So the reason why this is important is because the rotational freedom that you see here allows for the peptide excuse me, it allows for the peptide or the protein to rotate the position of these R groups, of these side chains. So we can rotate those side chains so we can position them so that they are interacting favorably with other side chains in the protein, or maybe with the solvent, which is probably going to be water, and so that we can avoid steric clashes. Basically, we don't want to get two atoms too close together, so we can rotate a lot of the atoms within the structure here so that atoms aren't getting too close together, because they'll want to repel each other if that is the case. Okay, so now let's talk about our 20 different amino acids. So all we really know at this point is that we've got 20 different amino acids, and what makes each one of them unique is the identity of their side chain. So when we end up synthesizing a protein, whether it's inside the cell or in an in vitro setting, if we do it outside the cell, the sequence of amino acids in that protein, depending on which order we put the amino acids together in building the polymer, that is going to impart a certain function to the protein based on the chemical characteristics of those side chains and how they like to interact with other molecules once the protein is finally folded. And we'll have a conversation on protein folding later on. So what we can do here, so 20 amino acids is kind of a lot to deal with, so let's start by classifying them. So instead of talking about 20 different amino acids, let's talk about maybe 5 or 6 different classes of amino acids. So the first class are what we call nonpolar or aliphatic amino acids, and they are very hydrophobic. We also have polar uncharged amino acids, which are hydrophilic, acidic amino acids, which are also hydrophilic, basic amino acids, which are hydrophilic, and then aromatic amino acids, which are hydrophobic. So those words that we just introduced, hydrophobic and hydrophilic, you can probably work out what those words mean, right? So let's start with the word hydrophobic. Hydrophobic literally means fear of water. Hydro, the prefix referring to water, and phobic or phobia referring to a fear of something. So if you've got an amino acid with a hydrophobic side chain, it means that that side chain isn't really going to want to interact with polar molecules like water. So if the side chain is nonpolar, it's going to want to interact with other molecules that are also nonpolar. And because water is very, very polar, that's going to be like mixing water and oil together, which is another example of the exact same chemical properties at work here. Hydrophobic molecules do not like to interact with hydrophilic molecules or water itself. So if you look at these different molecules here, these different amino acids here, take note of the side chains which are labeled here in pink. So you'll notice that everything else about these different amino acids is exactly the same. You've got the alpha carbon, you've got the amino group, the carboxyl group, and then just another hydrogen hanging out here. Everything that you see in black here is identical between these different amino acids. What makes each of them unique is their side chain, which is labeled here in pink. So you've got alanine, which has just a CH3 group here, valine, which has kind of a V-shaped aliphatic group here that contains just carbon and hydrogen here. Isoleucine, same deal, just a little bit differently put together. Leucine, which looks kind of like valine with an extra CH2 here. Methionine, which is mostly carbon and hydrogen with a sulfur atom thrown in there. And then you've got what we're also going to kind of classify by themselves as the aromatic amino acids here, phenylalanine, tyrosine, and tryptophan. So we'll talk about them a little bit later. But the main thing to take from here is that you're seeing a lot of carbon and hydrogen. So carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. So just carbon and hydrogen together, if that's the only thing that you find in a molecule, it is going to be nonpolar and therefore is not going to want to interact with water. So if you have a polypeptide that has a lot of these hydrophobic amino acids in it with all these side chains sticking out, this isn't going to be a polypeptide that's really going to want to be in an aqueous solution because these side chains are really not going to be too crazy about interacting with water. So if we look at some of the polar or hydrophilic amino acids, then obviously the word hydrophilic is going to mean exactly the opposite of hydrophobic. Hydrophilic literally means lover of water. Hydro meaning water and philic or philia referring to a love or an affinity for something. So hydrophilic amino acids are, in contrast to the hydrophobic ones that we just saw, they are going to have either very polar side chains or electrically charged side chains. And as such, these are going to be very, very highly capable of interacting with water in a good and favorable way. So these side chains are going to be very willing to interact with water that is surrounding your protein, whereas the hydrophobic ones are not going to be so crazy. They'll want to kind of hide away from the water. So the 4 amino acids that you see here, they are not the only hydrophilic ones, but they just happen to be the ones that do not have electrical charges. So we'll go ahead and start with them. You have serine, threonine, asparagine, and glutamine. If you look at serine and threonine, you'll notice that you have some carbon and hydrogen here, but you do also have a hydroxyl functional group here. So that's going to impart some polarity to the side chain and allow it to interact with water. Asparagine and glutamine both have amide functional groups. That's what you call the carbon, oxygen, nitrogen setup that you see here. They have amide functional groups, which are also very polar and very able to interact with water. Now, one note I will throw in here is that tyrosine, which is one of the hydrophobic amino acids that we were looking at right here, it is mostly nonpolar, but it is noteworthy that it does also have this hydroxyl functional group here, which really tells us that if it has to interact with water, we could probably get it to do so. So it has some polarity because of that hydroxyl group, but we're still mostly going to call it nonpolar. So if you really want to split hairs there you go. Okay, so continuing on with the hydrophilic amino acids, these are going to be the ones that are not only hydrophilic, but are also electrically charged. So the amino acids that have side chains that carry electrical charges at neutral pH values, these are also hydrophilic. So first we have what we call the basic amino acids. So this is going to include lysine, arginine, and histidine. So the basic amino acids at neutral pH values, so around 7 or so, they are going to carry positive electrical charges. So you'll see a positive here on the lysine, a positive here on the arginine, and a positive here on the histidine. So when you see the word basic, think positive charges. And then for the acidic amino acids, you'll want to think the opposite. They are going to carry negative charges at neutral pH values. So aspartate or aspartic acid carries a negative charge here. glutamic acid or glutamate carries a negative charge here. So when you see the word acidic for amino acids, think negative charge. Basic means positive, acidic means negative. So if you know anything about how positive and negative charges attract each other, it shouldn't surprise you that acidic and basic amino acid side chains are going to be attracted to each other via electrostatic interactions. So once we get to the point where a protein folds and we start getting side chains interacting with each other, acidic and basic side chains are going to have a high propensity to interact with one another because they like to interact with one another. Okay, so let's focus in real quick on histidine. Histidine is one of those basic side chains that we just talked about before. But histidine is a little bit different because the side chain has a pKa value of 6.8, which is very close to neutral pHs. The difference here is that the other basic side chains, those of lysine and arginine, have pKa values that are much greater than that, say around 11 or 12 or so. So if you don't remember from Chemistry 2, pKa refers to the pH value at which 50% of an acid or a base is in the form A and 50% is in the form HA, so either unprotonated or protonated. So if you look here, you're going to see histidine's side chain at two different pH values, both at 5 and at 7. So at a acidic pH like the pH 5, the side chain here is going to be protonated, meaning it has an extra hydrogen atom here, which is going to impart a positive charge to this nitrogen atom here. But as we start increasing the pH value, we are going to eventually pluck that hydrogen off and deprotonate the side chain here, such that this nitrogen atom here no longer has the proton and it is going to be neutral and no longer electrically charged. So the reason why we bring this up, and we're going to have a whole lecture on this very concept of side chains becoming protonated and deprotonated later on in the semester, so there's not really any need to worry about it too much just now. But because histidine has a pKa that is so close to neutral pH, it means that if the pH is 7, or let's just say if the pH is 6.8, that means of all the histidine side chains that we have in our protein, half of them are going to look like this, and half of them are going to look like this. So half of the side chains are positively charged, and half of them are going to be neutral. Whereas if you look at something like lysine, because its pKa value is something in the neighborhood of maybe between 12 and 13, a neutral pH is going to be so far below that pKa value that for all intents and purposes, 100% of the lysine side chains are going to be positively charged. So thinking about a lysine that is not positively charged really isn't even worth thinking about. But because histidine's pKa is so close to neutral pH, you really do have to kind of think about this. So the reason why this is physiologically significant is because inside of our bodies and inside of our cells, because all of our bodily fluids are mostly maintained at a fairly neutral pH, like our blood at 7.4, like the inside of a cell around 6.8 or 6.9, then because those pH values are so close to the pKa of histidine, this means that any protein in the cell that has histidines is going to have kind of a pretty equal proportion of histidines that are positively charged and histidines that are not positively charged. So that if inside the cell, if there is some kind of momentary change in the pH, whether it becomes more acidic or more basic, that's going to be very significant because it's going to change the proportion of how many of those side chains are positive and how many are neutral. And that can really affect the function of a particular protein if that histidine side chain is participating in, say, the catalysis of a reaction, if this is an enzyme that we're talking about, or it can have an effect on how the protein folds. So those are the sorts of things that we're going to eventually want to be able to think about. This is a pretty complex thing to talk about, the very first lecture for proteomics, so I wouldn't worry about it too much just now, but there is going to be a time this semester where you're going to want to be fairly comfortable with this, so better to go ahead and introduce it now if possible. Okay, and then finally let's talk about the aromatic amino acids, which we already showed before as part of the hydrophobic amino acids. So these are phenylalanine, tyrosine, and tryptophan. The one thing that you'll notice that these amino acid side chains have in common is they all have a phenolic ring structure. So you see that there with phenylalanine. It's basically the same thing with tyrosine. Tyrosine is pretty much identical to phenylalanine, except it also has a hydroxyl functional group attached there. And then tryptophan has not only the phenolic ring, but also a kind of a pentagon ring structure on top of it as well. But basically the thing they all have in common is that they've got ring structures. So these are worth mentioning on their own is because if you look at the rings, you'll notice that we kind of have these alternating double bonds. So double bond there, double bond there, double bond there, and the same and so forth. So I don't want to try to turn this into an organic chemistry class, but something that is significant to know about these ring structures where you have these alternating double bonds within a hexagonal ring like this is that the electrons don't just stay in one place. They are actually delocalized, meaning these electrons are constantly moving in a circle around this ring structure, and basically they generate something resembling an electrical current that runs throughout the ring. So the reason why this is significant is because you can actually get attractive forces between these rings if you stack them on top of each other. So aromatic rings like this are able to stack on top of each other and produce these special type of attractive stabilizing forces called pi-pi interactions. This is something that you tend to see and discuss a lot more when you're talking about DNA molecules, in which those purine and pyrimidine rings like to kind of stack on top of each other as you kind of wind up through the double helix structure. But this is definitely something that you can see happening when you get proteins folding, and you can get, say, phenylalanine stacking on top of each other, or a tyrosine stacking on top of a tryptophan, or something similar like that. Okay, so that takes us through all 20 of the amino acids, so we can pretty much categorize amino acids based on whether they are hydrophobic, meaning they're nonpolar, polar amino acids that are uncharged, basic amino acids, acidic amino acids, and then finally the aromatic amino acids. But there are a few things that we do want to say about some of these amino acids that we didn't get a chance to before. The first one we want to highlight is the amino acid glycine. Glycine is the smallest amino acid by far because it has the smallest side chain. Its side chain is literally just a single hydrogen atom, so you can't get much smaller than that. So glycine is going to be it's going to be by far the smallest and the lightest of the amino acids. Because it is so small, glycine can actually fit into very, very tight spaces within a protein structure, and that's going to be very useful when we start talking about folding the protein and getting the protein nice and compact into a small volume. Having glycines allows us to really get that protein compacted down into a small size. The amino acid proline is definitely an interesting one to talk about because you will notice here something very strange. The side chain is actually not all by itself. The side chain is actually fused to the amino group that is attached to the central carbon. So that's very interesting. So for all other 19 amino acids, the side chains just kind of hang off all by themselves. But for proline, the side chain is actually connecting the alpha carbon to the alpha amino group here. So it's very interesting, and technically proline, it's not even really supposed to be called an amino acid. It would be called an imino acid, but that's splitting hairs. For our sake, we still consider it an amino acid. So because of this fusion between the alpha carbon and the side chain and the amino group, proline is going to be very, very, very rigid. It's not going to have that rotational freedom that we talked about before. So this is going to form a fixed kink in the protein chain, and because of that limited rotational freedom in the vicinity, this is usually going to mark a place where a protein likes to kind of fold back on itself. So it's kind of like, if you think about like a wire coat hanger, think of the proline kind of being the place on the edge where you kind of fold that wire back so that you can actually hang a coat on it. And then another amino acid that we want to highlight is the amino acid cysteine. Cysteine has a free sulfhydryl group. That's what we call the SH functional group here. And this sulfhydryl group is actually capable of forming covalent bonds with other sulfhydryl groups, like those found in other cysteine amino acid side chains. So when you get a covalent bond formed between a sulfhydryl group here and another sulfhydryl group, that is an oxidation event. So when conditions in the cell promote oxidation over reduction, so promoting the gaining of electrons over the loss of electrons, this is going to lead to, in a lot of cases, the formation of what we call disulfide bonds within proteins, having a cysteine of one part of the polypeptide chain forming a covalent bond somewhere else where there's a cysteine somewhere else within the protein. So if we focus on this concept of disulfide bonds are actually very useful in that they can help to lock a protein into its folded configuration, basically to kind of lock it into place. So once you get a protein folded, you don't really want it to kind of fall apart on itself, so you can kind of glue it together, for lack of a better expression, by forming these disulfide bonds. So you're not going to see this as much for proteins that are inside the cell. You're going to see this a lot more for proteins that exist outside the cell because they don't have access to the intracellular chaperone machinery that helps to get proteins folded and to keep them folded. So you can kind of see what we're talking about in this diagram here. You have a protein that has a free sulfhydryl group here, so it has a cysteine side chain here, and then maybe another protein has a sulfhydryl group here, or maybe the same protein, just a different part of the same protein, has a cysteine group here. So when we get these positioned very close to each other, we can get an oxidation event in which we form a covalent bond between those two sulfur atoms, and that is what we call the disulfide bond formation. So whether we have disulfide bonds or not, it's going to basically depend on the balance between oxidation and reduction inside the cell. So you learned back in probably both General Chemistry 1 and General Chemistry 2 that a lot of chemical reactions are the result of the transfer of electrons, both oxidation and reduction. Oxidation referring to gaining electrons, and reduction referring to the loss of electrons. So whether conditions favor oxidation, in which you would see more disulfide bonds, or reduction, in which you would see fewer disulfide bonds, that's going to be a lot of what determines whether we form disulfide bonds or break them. Okay, so now that we've talked about our 20 amino acids, let's talk about another layer of complexity here. Let's talk about stereoisomers. So an isomer, something that hopefully you've heard before, an isomer refers to two molecules that have exactly the same chemical formula, but the atoms are attached in slightly different ways. So those are what we call isomers. They have the same chemical formula, but they're two different molecules that are attached in three-dimensional space. So stereoisomers are a type of isomer that is basically what we're dealing with here. And amino acids are stereoisomers, unfortunately for us, something that we have to kind of keep track of here. So the result of having two different stereoisomers like what you see here, so you've got your alpha carbon, your carboxyl group, your amino group, you've got your side chain here of CH3, so that means we're dealing with the amino acid alanine, and then also a hydrogen atom here. So we can actually make different types of molecules based on how these different groups are attached to that central alpha carbon. So if you look here, we've got two different types of the amino acid alanine because everything is attached in slightly different ways. So what we end up here is we have two different molecules that are non-superimposable mirror images of each other. So if you kind of use your imagination here, kind of imagine these molecules rotating. So start with the molecule here on the right. Imagine rotating it just a little bit to the right. Try to rotate it so that this NH2 group gets to about where the NH2 group is right here. So you'd probably rotate it maybe about 45 degrees to the right. Okay, so if you do that, you'll get this NH2 group positioned in the same way that it is over here, but where the CH3 group is here, that's going to be where the hydrogen atom is here, and the CH3 group here will be kind of back here at the back of the molecule. So basically what we end up with here is that these are two completely different molecules because you cannot rotate these molecules in any way so that these functional groups are positioned in the same way. So these are two completely different molecules, even though they look like the same molecule, they contain all the same atoms and the same functional group, but because these functional groups are attached to that alpha carbon in different ways, they end up being completely different molecules. So for stereoisomers, like what you see with alanine here, we name the stereoisomers with the letters L and D. So what we have here on the left is we have what is called L-alanine, and on the right we have D-alanine. Again, they look like exactly the same molecule, but because they are not superimposable with each other in three-dimensional space, they are not actually the same molecule. However, if we could actually make them become the same molecule if we just swap the position of the CH3 side chain and the amine group here. Then they actually would be exactly the same molecule, but since we can't do that, unfortunately they are not the same molecule. So the reason why stereoisomers like alanine and like other amino acids exist is because amino acids, and not just amino acids, but also monosaccharides and other types of molecules as well, they contain what are called chiral or asymmetric centers. So a carbon atom is referred to as an asymmetric center if it is bound to 4 different chemical groups. So look at alanine here. You've got your central carbon, amino group, carboxyl group, hydrogen atom, side chain. So this is a asymmetric chiral center because this alpha carbon is attached to 4 different things. This is unique, this is unique, this is unique, and this is unique. So those are 4 different types of things that are attached to that central carbon, meaning that how we actually position those things around the alpha carbon is going to impact what that molecule actually ends up being. So of the 20 amino acids, there is only one of them that does not exist in both the L form and the D form. So without looking any further, can you figure out which one of those amino acids it is? So you have to think about the side chain. So if you want to be spoiled with the answer here, the answer is the amino acid glycine. Glycine has a side chain that is just a hydrogen atom. So because the hydrogen atom is the same as the hydrogen atom over here, the alpha carbon in glycine is actually not an asymmetric center because even if we start swapping the positions of these groups here, it's not going to matter because these two groups are identical to one another, so we can't make stereoisomers for glycine because there's no way to end up with 2 different molecules that are non-superimposable mirror images of one another. So glycine is the only amino acid that does not have an L form and it does not have a D form. There is literally only one kind of glycine, the one that you see right here. However, for the other 19 amino acids, if we were to swap the positions of 2 of the groups attached to that alpha carbon, it would definitely change the molecule in 3-dimensional space, and the 2 stereoisomers, like we said before, would become non-superimposable mirror images of one another, and they would become distinct molecules. So what you're probably thinking at this point is, man, what a pain in the ass, right? So not only do we have 20 different amino acids, 19 of them actually have 2 different forms. So you could almost argue that we don't have 20 amino acids, we've got 39 amino acids, right? So what a pain in the rear, man. Well, here's the good news. The good news is that because the L and the D forms of these amino acids, because they're not identical molecules, it probably makes some sense that the cell and the cell machinery probably has a preference for one over the other. So as it turns out, proteins produced in the cell and proteins used by the cell only are going to use L amino acids. So D amino acids really are not really worth thinking about for us because living cells do not use D amino acids for much at all. So if you're thinking, man, what a pain in the rear having to deal with not only L amino acids, but D amino acids too, it's good to understand the whole stereoisomer concept, but L amino acids are all that's really going to matter, so it's really all you're going to have to think about. So we go from 39 amino acids back down to 20, so that should help you out quite a bit. And even though this is a proteomics class, something that we can go ahead and throw out there as a bit of trivia is that in the same way that the cell uses only L amino acids, it turns out that the cell actually only uses D monosaccharides, with L sugars not really serving any biological purpose. So D-glucose, D-galactose, and other monosaccharides, D-mannose, those are the only ones that the cell uses. So L-glucose, L-mannose, L-galactose, those really don't have any biological purpose. Okay, so there is a reason why we want to be aware of this. So when we're doing our lab project for the semester, so if you haven't come to class for the first time yet, we're going to talk about the project that we're doing this semester, in which we are going to try to isolate, purify, and characterize the enzyme tyrosinase, which we can get out of a variety of sources like fruits and vegetables and things like that. The enzyme tyrosinase has a substrate called L-dopa. So L-dopa, because it starts with the letter L, this should tell you that it contains an asymmetric center, which I have highlighted here with a red circle. So as an asymmetric center here, L-dopa also comes in another stereoisomeric form, D-dopa, where you can see this NH2 amino group actually can either be coming up out of the plane of the image here, or it can be projecting back. So these are not identical molecules here because of the positioning of the functional groups here. So what we're going to eventually do this semester is once we have our purified tyrosinase enzyme, we are going to test whether it has an affinity for L-dopa or whether it has an affinity for D-dopa. Can it catalyze reactions with one better than the other? So it might be a good idea for you to go ahead and think about, will tyrosinase catalyze a reaction more efficiently with one substrate versus the other? Or maybe it doesn't have a preference. Just something for you to go ahead and think about. Alright, so that is going to do it for this first video. It was kind of a long one, so hopefully most of these videos won't be quite so long. But I thank you for your attention, and I will see you next time. So long.